video with Mr. Phillips on A-level biology. Today we're going to be looking at the whole of the immune system, the natural immune system anyway, or natural active immunity, because it's quite a big section. I'm going to do this all together though, because I think it works well if we go through it from the exact start to the exact finish. You might want to pause this video at several places just to make sure you've got all the information down, and I'd strongly recommend use the book because I might miss little bits out obviously going through it. So let's start off then. So we start off with a picture on there, which is a macro page. Let's have a look at what we're going to be talking about. Basically, first of all, you need to know what an antigen is. Okay, so hopefully by now you already know that an antigen is a large foreign molecule stimulating immune response, including the production of antibodies. Okay, so the key thing in there is it's a molecule that stimulates the immune response. Normally it's foreign. I don't like that word particularly because it doesn't necessarily come from outside your body. It might be a cell that's mutated in your body that's got these antigens as well. It's normally a protein. Okay, so you could say glycolipid or glycoprotein as well, but normally a protein. And basically our own body cells also always have antigens, but they're self-antigens, so we don't normally trigger an immune response against our antigens. The other things that will have antigens will be, as I said, obviously pathogens, bacteria, cells that have mutated, or even toxins of bacteria might have antigens on them. So this happens then, obviously antigens are very important. It helps to recognize the immune, the response we need to do to create antibodies. But there is something called antigenic variation. And this is where you have different strains of viruses or bacteria. And it's due to obviously mutations, because bacteria and viruses and pathogens have high, fast life cycles, quick life cycles. So therefore they've got a much higher mutation rate than we will. So what happens is obviously their DNA changes, that changes the amino acid sequence, and that will alter the antigens, so they no longer work. So vaccines always are used using the most recently circulating viruses. For example, if you're having the flu jab. An example of a disease where you don't have any immune system, where you have a weakened immune system, is severe combined immunodeficiency, which is called SID. You have your primary defenses, so what that means is that you will have like your stomach acid, your skin, things like that, but your actual white blood cells don't recognize the foreign antigens or don't recognize them as well, and that therefore is an issue. And this is an example of someone who had SID, was known as the ball boy, and obviously it's obviously very limiting to your lifestyle. Okay, but today we're going to be looking at the immune system in a sense. So first of all, you've got your primary defenses. Your primary defenses are the ones that are stopping the pathogens getting into your body. So it could be physical barriers such as skin, it could be chemical things such as lysozymes, or it could be even the T lymphocytes that are going to start facing. So start off with, then we're looking at phagocytes, which are part of the non-specific immune response, and this makes about 60% of your white blood cells. They've got multi-lobe nucleus, and they're made in the bone marrow, and then they go into your tissue fluid and around your blood to find invaders. They don't live that long because they are short-lived, and obviously they become antigen-presenting cells, and they're found a lot on the epithelial surfaces, such as your lungs, where obviously is a weaker area for bacteria to enter. So what happens then in the process of phagocytosis? Well, basically, in phagocytosis, you have this sort of series of events happening. So you start off with the bacteria or the pathogens. So there you go. So there it is here. And the phagocyte moves around it and engulfs them. The phagocyte cell membrane then fuses and it encloses the phagocyte into a vacuole around the bacteria. This is called a phagocytic vacuole. And then what happens is the lysosomes, which contain the enzyme lysozymes, and the lysozymes are hydrolytic enzymes which will break down the gram-positive wall of the bacteria, break down their peptidoglycanase cell wall. That is then fuses with that vacuole, and it breaks down the bacteria, it digests the bacteria. They then take the antigens of the bacteria cell, and the phagocytes display them on their own surface. So the bacteria cells antigens will end up becoming on the outside of the phagocyte, and that is becoming an antigen presenting cell. and that is what stimulates the T cells, all the B cells, to recognize the bacteria has invaded the cell and recognize it as an antigen-presenting cell, and that triggers the specific immune response. So again, just a different way of showing it here. So you can see again the phagocyte is attracted by chemicals that might be released by the pathogen. It attaches, engulfs the pathogen into its own vacuole, and then it releases the lymphocytes, lysosomes, sorry, release the lysozyme enzyme to break it down, and it becomes an antigen-presenting cell. So. And just to summarise, if you had a question in the exam, this would be a pretty much a perfect answer for it. So pathogens produce chemicals and other toxins that attract the white blood cells. Phagocytes recognise the foreign antigens on the pathogens not being of that organism. The phagocyte engulfs the pathogen, putting it in a specialised vacuole called a phagosome or a phagocytic vacuole. The phagosome combines with the lysosome, which contains powerful digestive enzymes called a phagolysosome. And then the enzymes of the phagosome break down the pathogen, and the pathogen's antigens are then presented on the phagocyte, making the cell of its cell surface membrane, making it an antigen-presenting cell. There is one other thing I think this is worth mentioning. A lot of the phagocytes live in your lymph nodes, which is where your neck swells up when you get ill, or your glands swell up in your armpits. It's not in the blood, and this is why if you get injury, you release a lot of histamine. So let's say I cut my hand would swell up. That is histamine, and what that does is it makes your blood capillaries more leaky, which means any bacteria that gets in is more likely to go into the lymphatic system, and that is more likely then to come across a phagocyte. Okay, so then let's go on to specific immune system. What we've said so far is non-specific phagocytosis occurs and they become antigen-presenting cells. So what do we do with those now? Well, how do they then trigger this actual specific response to kill off specific bacteria or specific viruses or other pathogens? Well, basically, it's specific as that means attacking one type of pathogen, and because it's got one type of antigen. And basically, the cells don't have lysosomes in, so it's not about releasing chemicals that are going to digest them. It's about actually coming up with new methods. So there's two levels we're going to look at: beta cells or B lymphocytes, and they form and mature in the bone marrow, and later they differentiate into something called plasma cells. They are the things that manufacture our antibodies. So they're obviously absolutely critical that they manufacture the antibodies. And this is often the question you get at A level: is how does the presence of a bacteria lead to antibody production? And that could literally mean you start from talking about phagocytosis to T cells to B cells. Okay, but they turn into plasma cells to manufacture antibodies, and they also become beta memory cells. You then got T lymphocytes or T cells, and they are formed in the bone marrow but mature in the thymus gland. And there's a couple of different versions which we're going to look at as we move forward. So let's start off with the T cells. The reason starting off with the T cells is because they are the things more likely going to detect the antigens on the antigen-presenting phagocyte or the antigen-presenting cell. And if we have a look at this example here, so they've said it on your body's own cell, but again, that could be a phagocyte, okay, or otherwise known as macrophage. And what happens is the T cell detects it using its surface receptors, which will have complementary shapes to the antigen, so it will detect the antigen. That causes then a trigger of several things. It causes something called clonal expansion, which is mitosis, to make lots of copies, and then they differentiate into one of these following types of cells. So first of all, the memory cell, that is one that just remains in the blood, and if it's re-exposed to the same pathogen again, it will produce it much more quickly and rapidly divide to create the response quicker, so you probably won't get ill again. Helper T cells, we'll talk about in a second, so don't worry about that, but it causes lots of other immunological steps. Cytotoxic T cells, cyto means cell, so cell toxic, cytotoxic T cells destroy the tumor cells and any host cells that are infected by the virus. So they will come along and they'll destroy any infected cells and any tumor cells, obviously, because we could run a cancer cell that's got cancer markers or mutated antigens on it. So that's the job of the cytotoxic cell. And then we've got the suppressor T cell, and this moderates the immune response. When all the numbers of antigens, foreign antigens have died down, i.e. you've killed off the virus and you've all the bacteria and you've killed off any infected cells, the suppressor cell then shuts the whole process down. Let's have a look at helper T cells then. So what's the importance of these? Well, the importance of these is that they basically cause the whole rest of the immune response. So they basically cause the maturity of beta cells, which will make antibodies, which we'll talk about in a second. They form or stimulate the formation of beta memory cells, which is obviously important for the next time, the second exposure, if you get it. They activate something called the cytotoxin cells, and they activate further failure sites if needed to fight the disease. So let's have a look at the beta cells. So first of all, the beta cells response. I should have really said the T cell response is called the cellular response. The beta cells are called the humoral response. And basically, a beta cell is a cell as a cell surface membrane that is covered in antigen receptors or antigen or antibodies, effectively, that are complementary to the antigens. And when they bind to an antigen, they will then trigger a response, and that, or it could be a T helper cell triggering the response, and that causes the beta cells to become activated. So what do the beta cells actually do? Well, each day we randomly make millions of beta cells with unique receptors on the surface. If a pathogen enters the bloodstream and one of the beta cells has a complementary receptor to it, it triggers this response. So what happens first of all is you get something called clonal expansion. So what will happen is the cell detects the pathogen, okay? That causes clonal expansion, which is mitosis. You must remember to say the words mitosis. And if it's an essay, you can then talk about what mitosis is a little bit. And then once you've got lots of numbers, you then do clonal selection, which is differentiation. And most of the beta cells become plasma cells, which float around the blood, and they make antibodies. Some of them become memory cells, which also remain in the blood, and they will be able to rapidly divide to make new plasma cells if the bacteria or the fungus or the virus, the pathogen, comes back into your body and it detects the complementary antigen once again, which explains our immunity to disease once we've been ill. So if we have a look at the whole of the immune response together, it sort of shows how it all works. So the first thing is you get the lymphocyte, which is, the phagocyte, which is digesting and engulfing the bacteria. That then becomes an antigen presenting cell, which then triggers the T cells to recognize it. Now, the T cells on this list are slightly different from the others, so don't worry about the actual names of T cells, but effectively that causes your T cell response. And then we have the T helper cells, as you know, which release the chemicals, which are called interleukins. You don't need to know that. That activates the beta cells to cause their response. And then obviously the other T cells take care of the cytotoxin cells, which are called killer cells here, destroy cells, and then obviously you've got the other cells which we just talked about. The beta cells then are activated by the T cells, and that causes clonal expansion. Again, that is mitosis. Then you get clonal selection, which is differentiation, which is some of the cells turn into plasma cells and they make the antibodies, and some of the cells turn into memory cells to give immunological memory for the further exposure in the future. Now, what are we actually making? Well, we're making antibodies. So these are, as we said, made by the B lymphocytes, by the plasma cells. What are antibodies? They are glycoproteins. You probably just get why I'm saying proteins that are called immunoglobulins, and they are released in response to a non-self antigen to form an antigen antibody complex. And you need to know the structure of a antibody in quite a lot of detail in order to answer an exam question on it, because you might guess how they work or what their structure is and how they work. So if we have a look, this picture on the right hand side is probably more sort of like an exam question would be, without the labels effectively. So the structure of a antibody is of this. You have something called a light polypeptide chain. Okay, so you have two of those. There's one there and again one on the other side. So it's light because it's shorter. Think of it like that. Then you have two heavy polypeptide chains. So there's one there that I'm drawing with green right now, and then obviously one on the other side. Now the other diagram then will have to just help us a little bit out here. Those polypeptide chains are joined together by disulfide bridges, and obviously you might have talked about those in the tertiary structure proteins before. Now, At either end, then we have slightly different things. So we have something called a hinge region, which allows some flexibility in the Y-shaped protein. You can call it a Y-shaped protein, and the hinge region allows the antibody to hold more than one protein, one pathogen at any one time. And then we've got two different areas. So we've got the constant region at one end of the pathogen, and then we've got the variable region at the other end of the pathogen. The constant region is this region here. So I've got a poor arrow. And the constant region basically is where the white blood cell, the phagocyte normally, will attach to the antibody, and then it will be able to absorb the antibody with the bacteria attached to it on the other side. The variable region is variable to the different types of antigens. So there'll be one variable region, let's say, for COVID-19. There'll be one variable region for chickenpox. There'll be one variable region for the antibody that deals with the flu. So every different disease will have its own antibodies with a different variable region, and that's where the antibodies are obviously being attached to the antigen. So it's complementary to the antigen of that specific disease, and that will be dependent upon obviously the amino acid sequence, which will then give the secondary sequence, which will then give the third tertiary level of the protein. So what do you need to know? So you need to know you've got a variable region that binds to the antigen to the complementary shape due to the amino acid sequence, and that will be different. It's specific to each pathogen. There is a non-variable region, which is the same, and it always attaches to phagocytes. And then you've got the hinge region, which allows flexibility, so it can attach to more than one antigen at a time. So how they actually work, how they actually kill off the bacteria or the virus or the pathogen, well, they do it by something called agglutination. So what agglutination is that the antibodies can stick to the surface of multiple microorganisms and cause them to clump together. So here you can see quite a nice picture of that. If they're clumped together, then they will no longer be able to infect you because they won't be able to work as efficiently in terms of viruses. They won't be able to get into your cells. Okay, but also then what that means is then they're clumped together, ready for a pathogen, for a phagocyte to come along and do phagocytosis, for a neutrophil to come along and do phagocytosis. So they'll act as a marker, and then what will happen is that the big phagocyte will come along and it will be able to attach, say, to this antibody here, and then it will engulf that antibody and all the bacteria or fungus attached to it. So that is how it sort of actually kills it off in the end. And the last thing is, well, you've got primary and secondary immune responses, and this obviously links really well to vaccines because it's how a vaccine works. But let's just talk about natural illnesses, active natural immunity to start off with. So you've got the initial exposure, okay, which is when we go through those stages of events we've just talked about. So you have your phagocytosis occurring, that activates your T cells. They do clonal expansion and clonal selection, which is mitosis and differentiation. You then get your T helper cells, which send out interleukins to the beta cells. The beta cells then do mitosis and clonal expansion. They do them differentiation to make the plasma cells, and they make the antibodies. Okay, so this graph shows you the antibodies, but that will take obviously a certain amount of time. So you get ill and you feel poorly for the first bit, okay, as your antibody levels increase. That then kills off that illness, and you don't feel ill anymore, and your antibody levels drop. And then if you get exposed the second time, you get a much more rapid and a higher production in number of antibodies in your secondary immune response. And that is because the secondary immune response, those beta memory cells are floating around, or the T memory cells, if it's that detects it floating around, they detect the antigens, the foreign antigens of the bacterial pathogen sooner, and they trigger that immune response at a much greater rate. So therefore you get no more symptoms, because it doesn't take half the time that it would for you to normally develop the antibodies on the first case. And notice the drop in antibody levels out, and it's quite slow. In fact, this graph I would say is a bit inaccurate. I'd say probably more sort of like that. So it takes longer to curve off afterwards. So that's specific immune response. So you need to know all the different stages. You need to know what's specific, what's non-specific, what the beta cells and T cells do, and then how that affects you in the primary and secondary immune response when you get infected a second time. Cheers, guys. I'll see you again soon.